This is another example where I was working at Swansea University. They have utilized waste tea leaves as a biosurfactant in cadmium sulfide quantum dot synthesize to study their cytotoxicity effect in breast cancer cells. But we need a high resolution image to identify. So these are the quantum dots, uh, cadmium sulfide quantum dots, which we identified through the uh, high resolution TM. Then later on, they have used it for imaging purpose with the fluorescent imaging like this, fluorescent microscopy imaging with the different treatments with the different concentration of MCF cell line. So this is how the fluorescent imaging can be used to identify the cytotoxicity effect in breast cancer cell. I was partly worked only with the microscopy image here. So this is another example where we got a curcumin loaded core shell zinc oxide silicon dioxide nanostructure for bioimaging and cancer therapy. Because the photo enhanced zinc oxide nanoparticles can produce a reactive oxygen species that leads to oxidative stress in a cancer cell. And zinc oxide nanoparticle can form negatively charged zinc oxide ion and positively charged zinc hydroxide ions that lead to an electrostatic interaction with the cancer cells and causes phagocytosis and cytotoxicity. So due to these surface defects, zinc oxide nanoparticles gives rise to a visible fluorescence. Porous silicon dioxide nanoparticle facilitates better surface functionalization through covalent bonding or electrostatic interaction and drug diffusion. The selenol groups present on the surface of silicon dioxide makes it hydrophilic in nature. So these hydrophobic drugs are binds with silicon dioxide pores structures by hydrogen bonding and electrostatic interaction, which improves the drug delivery system. So that's why we need a zinc oxide. That's why we need a chemical property of the positivity of chemical property of zinc oxide and silicon dioxide together. Zinc oxide is for enhancing the visible fluorescence effect, and porous silicon dioxide is for electrostatic interaction with the drug diffusion. So that's why we need it. So this is how we imaged in microscopy. We identified zinc silicon dioxide as a shell and zinc oxide as a core particle through high resolution TM images. So this is how the schematic looks: the zinc oxide and zinc oxide as a core and silicon dioxide as shell. The curcumin is loaded. The curcumin is used as an anti-inflammatory drug because we are using this material for cancer therapy. So we need an anti-inflammatory drug, chemotherapeutics, antimicrobial agent. So that's why we loaded curcumin with this material. The curcumin is effective against various type of cancers like leukemia, or breast cancer, or lung cancer, etc. Curcumin inhibits the proliferation in various phases of cell cycle and promotes apoptosis. It suppresses the transformation and metastasis of cancer. So that's how the material is used and tested for various like cancer treatment effect or cancer therapy effect. Of course, that fluorescent emission is used as a cell imaging together, like after the cytotoxicity effect. So this is how the imaging and drug therapy is combined together in this paper. So this is another example of how a scanning electron microscopy and transmission electron microscopy is used together to identify or to derive information from the virus particles, like different viruses like a salmonella or vaccinia or ebola viruses. So this is an ebola virus of imaged in sputter coated secondary electron scanning electron microscopy. Then this is one of the biological imaging process in SCM and TM using ionic liquid and negative stain imaging to get more information compared to the sputter coated as a conventional method. So this is how transmission electron microscopy is used, for example, for the Ebola virus and for the vaccinia or salmonella. It gives more internal information compared to scanning electron microscopy, either with this sputter coated or with ionic liquid. So these type of samples can be visualized in microscopy. This is one of the HNN1 infected cell, the antiviral activity of methylated beta lactoglobin. So this is a different activity stages which is imaged in transmission electron microscopy. For example, this is an HNN1 infected cell. Then later on, the methylated beta lactoglobin is introduced. This is how the activity is conducted or gone through. So the final stage of complete antiviral activity of this liquid together with this HN1 virus can be imaged in transmission electron microscope here. So this is one of the coronavirus imaged in transmission electron microscopy. It's a SARS-CoV. I think this was the first image from the last year, and this is another image from Hong Kong University, which shows a new coronavirus spreading in China, and several other countries uses the same cellular protein as SARS to gain access to cells. So this is one of the image which shows how these proteinated virus particle which acts or uh, process their element into the human cells. So this is one of the image which derived from the China virus. Oh, sorry, this is China virus, China image, and this is a Hong Kong image. So this is a microscopy image of cell infected with a novel coronavirus grown in culture at the University of Hong Kong. A multiple virus particle here are being released from the cell surface here. If you look into here, then it later on it develop its activity with the human cell here. So this is how the biological things or bioimaging through the electron microscopy. So this is another virus element which was imaged in scanning electron microscopy. That's a Viro E6 cells infected with severe acute respiratory syndrome, which is more or less similar to what coronavirus can make. That's a acute respiratory syndrome that's imaged at 24 hours after infection. A cell surface is covered with an extracellular progeny virus particle, and the progeny virus are being extruded from or attached to numerous pseudoporia on infected cell surface. If you look into the B image, a higher magnification micrograph of virus clustered pseudopodia, which shows in arrows. So then those matured regions are imaged in scanning electron microscopy here, which shows the form and structure of virus with a negative staining. That's an inset. This inset is from transmission electron microscopy here, which was imaged from the coronavirus infected cell after 24 hours. So this is how the transmission electron microscope is used for biological applications. So let's see. I will just try to give some information, like what are the advancement we can do after this imaging. So one of the technique, that's artificial intelligence or machine learning, which gets more signals or which gets more information through the processing of different or advancement in the softwares or programming. So it's an underlying concept of combining physics and mathematics together. So these are the advancement in healthcare system. For example, this is one of uh, the the process which may be developed in future or into the application in future. It There are advanced healthcare system which is associated with the human body, uh, which calculate all the things. Then this network is used to get more help through the medical server or through the emergency or caregiver or sending a doctors. So this is one of the approach in a modern world, that's now which is getting combining microscopy and machine learning in healthcare application into the next stage. For example, before deriving or before going into this structure, uh, imaging is done from the human cells, which is coming for a medical treatment, or the lab reports, together with the clinical data and diagnosis report, which is now further enhanced or further developed through the machine learning from the clinic's input. So those deep analytics can be done through the different data sets which is developed from machine learning or artificial intelligence. Then it chooses which type of treatment is needed from these lab data itself, with certain inputs from the clinics or physician. So this shows how much or what type of treatments needed, like whether it is just an exercise or drug choice or chemotherapy or surgery, which predicts the future from the imaging alone. So the world was focusing to this broad criteria, with an advancement in the signal processing, with an advancement in the embedded system, with an more advancement in the micro and nano processing system. And before that, now the Singapore is introduced such system, which is now in an operation only within lab imaging or lab reports. Through the machine learning advancement, anything can be processed. So this combines a physics, a biology, and mathematics or softwares. So this is an ongoing development together with imaging and microprocessing. So this is another digital image processing techniques which is developed recently from the image what you are getting, either from the virus image or from the material image. So let it be whatever the type of images you derive from transmission electron microscopy or any type of microscopy. So now the Python is used for the further image processing, for further scientific analysis through the machine learning and deep learning process. So which have shown here, like images acquired from microscopy. Then there are some pre-processing and post-processing analysis through the MATLAB and Python. Those analysis can be achieved later on for the further analysis and simulations. So the steps are like the microscopy consists of 5 main sub packages. That's including processing, that's a physical model independent processing of data, including a statistical learning, a image processing, and signal filtering. Then the analysis stage, which comes in with a physical model based analysis of data here. Then the simulation modeling of physical phenomena, then the interactive widgets and plotting tools specific to classes in processing, analysis, and simulation. So these are the advanced image processing techniques which also combined here with the lab reports, because the lab report also have imaging from the either from PET imaging or from the CET imaging or from the laboratory microscope imaging. So these are the tools used in future to get as much as more advancement into the need. So this is another example from digital image processing, which combine Python and machine learning, which also use mosaicing from the C++ and image J from the Java and Foresight from C++. So these cells are reconstructed, and the image analysis and image visualization can be further done by the deep learning techniques. So this is how it explained from the like profiling of brain tissue surrounding implanted neuroprosthetic devices. So those data sets are imaged here. That's how it's combined with the softwares and cell reconstruction and image analysis. So there are some technical terms which shows that resulting images typically contain 5 fluorescent channels. So each channel consists of 6,000 cross 10,000 cross 500 voxels with certain bits like pixels. which implying image sizes exceeding 250 GB. So these images must be mosaiced here, pre-processed to overcome imaging artifacts. So those pre-processing can be done from image J. So then the segmented to enable cellular scale feature extraction. So that's done by cell reconstruction and uh, image analysis. So these are the steps now followed using advanced field. For example, here the cell nuclei and microglia through the similar re- processing or image processing technique. So those specific image can be like enhanced unmixing and mosaicing. Then the nuclear segmentations can be done, the nuclear cell type identification. So these are the data which is already uh, derived from these techniques. Like there are some set of files, there are some set of analysis, there are some set of archivals. So those deep learning techniques which derive information from previously bounded information from the new information, which compare all set of data with that's why we call this an artificial intelligence. So this classification can be done with the interactive analytical tools to identify cells in detail. So for example, image shows a multi-channel raw data here. B shows a flowchart of how it it processes the images, and this shows how the final reconstructed image looks like. So this is I wanted to show you more than imaging from transmission electron microscope. There are advancement in the field. You have a lot of thinking process now to get either from the microscopy perspective or from the microscopy perspective, either from materials or from the biological perspective. So wherever you're working, whether it is chemistry or physics or mathematics or from the computer science, now now this world is an interdisciplinary and combined world to work together to get information from the cell imaging or from the material imaging or from any tools. So these new tools from the Python and machine learning is used to get development from your own field to the other field. So that's why I'm saying the world is interdisciplinary and advanced. So this is how what we combine from imaging and imaging from microscopy and the machine learning. So I just wanted to show you a few things, just a few final slides, how the material science is now used into the bioimaging. I just take one example. I don't want to get broader approach. I just want to introduce a carbon nanotubes. The carbon nanotubes or allotropes of carbon, which is more or less a cylindrical part, which is like folded and form a tube like this. A single layer of graphene looks like this, and multi-layer graphene is folded as a multi-layer carbon nanotubes. So these carbon nanotubes have wide choice of biomedical application, either from therapy or drug delivery or imaging and diagnosis. From the imaging, you have a lot of wide choices, whether it is MRI, multimodal imaging, or PET, or fluorescence microscopy. So this material is now further developed in different a combination. For example, here is a single one nanotubes together with a gold particle or the silver nanocomposites. So these single wall carbon nanotube with the gold and PEG nanocomposites, it looks like here, and the TM images and scanning electron microscope images looks like this. This is now used for cell imaging or fluorescent imaging. We can also use as a Raman imaging tools. So this is one of the live mouse image which was infected. So the injection was done through this carbon nanotube with a polyethyl glycolated single wall carbon nanotubes taken at different time points of post injections through the lungs, through the kidneys, and through the liver, spleen, lungs. So which identify the infected regions using fluorescence imaging or Raman imaging can be used. So this is one of another example where it shows the PET image of tumor bearing mice. So this is an MR imaging of stem cells tracking. So this is a Raman imaging of cells and fluorescent imaging in a different regions of different locations within a body. So for all these things, now the carbon nanotube is used as a drug delivery system, as an imaging system, or the carrier with the nanocomposites. So that's why I just wanted to introduce this sample or material, which is now used in advanced bioimaging tools. So with this one, I'm concluding my talk. If you have any questions, I think we have few minutes left out. We can go through that.
Thank you, Doctor. Thank you for your wonderful presentations and your presence in BioYojana 2021, organized by Tanway R&D Solution. Now, this is a section is open to the audience. Audience, if anyone like to ask question to Dr. Vijay Shankar Asokan, kindly raise your hands. Dr. Kumaran Shanmugam, now you can ask your question to Dr. Vijay Shankar, sir.
Thank you, sir. This is only appreciation. Then I came very late. I'm sorry, I had some meeting. So it's a wonderful presentation, sir. It is going to be for the youngsters, for the researchers. They can take the message, and it will be like permanent record where you are specialist in microscopic imaging, and you also touched like viruses and other types. So I'm very happy, sir. So thanks for your presentation, and thank you very much, sir. So I hope you are reached Sweden now safely, and you are there in Sweden. Is it right, sir?
Yeah, I'm in Sweden now.
Okay, thank you, sir.
Find work after 7 days quarantine.
Thank you, sir.
Now, anyone wants to ask question to Doctor, kindly raise your hands or give a chat in your box.
Maybe Purushothaman, I would like to. I came very late, sir. It is going to be very basic one, which I would like to ask you because I have not also experienced scanning electron microscope. You are a specialist in this technique or tool. So when we do select a sample, and when we go to the certain phase, what are the precautions one has to take? Like samples, what kind of nature sample is better for SEM because its electron is going to bombard. So if it's something like that, could you tell some for youngsters to when they are going to work, it will be useful.
So it's depends on first identify the sample nature. For example, it's whether if it is a material based, so we need to identify there is a conductive sample or the non-conductive sample. So if it's conductive, we can directly take it. If it's a non-conductive sample, then we can do some sputtering like gold or something like that. And if it is a biological sample, then we need to prepare separately, like liquid cell, ionic liquid phase, or through the negative strain. There are wide choices of biological samples to be imaged in scanning electron microscope. So once the sample preparation is done, so identify the tool what you need, whether you need a secondary electron imaging or the backscatter electron imaging. The secondary electron imaging, it's just for the morphological identification. And the backscatter electron imaging, which sometimes identify the different regions with the different brightness and contrast. This is more helpful in metal-based or metal alloys. And particularly for the bombardment, you need to be very careful with the sample's nature and the electron beam power. So for the biological samples, you usually go for low kV imaging, like 5 kV or less than that. And for the metals or materials, alloys or composites, you go more than 10 kV. So you just need to first study the sample, then identify what type of image, then identify how much I can bombard, and or sometimes I may need higher kV, but the sample cannot withstand for a long time. So you need to go for quick imaging. So it depends on you need to have some more study of your sample before going to image. So that's more needed.
Thank you
So
very much.
anyway, I have explained all these things in my YouTube channel. I have introduced during my talk. So all these things in detailed with the different directors and different setups I have explained there.
Great, sir. Thank you very much.
So if anyone like to ask question to Dr. Vijay Shankar Asokan sir, kindly raise your hands. Sir, I have one question, sir.
Yeah.
What is the importance of splattering and what's the process splattering?
Sputtering, is it?
Splattering, sir. In SEM ACM, use splattering for the biological samples. Sir, is this audible?
So most of the biological samples are non-conductive. So you need a sputtering to get more secondary electron enhancement. Otherwise, you don't get imaging, or sometimes you get charge building and get blurred. So you need a sputtering like either carbon or palladium or gold. That's a conductive material, which coats around 5 nanometer to 10 nanometer or 5 nanometer like that. So you get more secondary electron enhancement just for the imaging purpose.
Thank you, sir. One participant asked the question, sir. Her name is Shivani Yadav. What he said is, good afternoon, sir. As you said, carbon nanotubes have multiple uses as for drug delivery and in therapeutics. So, sir, my question is, carbon nanotubes, are they made in lab means? Can they be manufactured easily, or it's difficult process?
No, there are very, very simple techniques nowadays available to synthesize carbon nanotubes in labs. I personally did a PhD in nanotubes alone, so I have come across different techniques from thermal CVD or from the microwave oven or from the chemical route. So there are many techniques available. You can get it from your lab. If you have CVD, then it's very easy. If you have just thermal evaporation or CVD techniques.
Thank you, doctor. Thank you for your answer. And she also sent the thank you message, sir. Thank you. If any other likes to ask question, Soundarya CK. Soundarya, now you can just switch on the speaker and ask the question directly to you, sir.
As nanotechnology student, where will I get hands-on training in electron microscopy techniques?
Soundarya, now you can directly ask the question to sir.
Sir, thank you for your wonderful presentation, sir. As a nanotechnology student, where will I get the hands-on experience in electron microscopy techniques or any other microscopy?
We have offered different courses even from my side, but those are all very theoretical microscopy courses. But we are also trying to get hands-on training, because that's where we are lacking on, from at least from the Indian perspective. We have a lot of like many instruments available throughout India, but it's not frequently used for training purposes. So if any institute is ready to provide that, even we can arrange some sessions. I don't know whether there are already institutes are offering the hands-on training sessions. I'm not sure about it.
Okay, sir. Thank you, sir.
Sir, just I would like to add a point, sir, for you. Indian Institute of Science probably is a nanotech student. There is a center, they have a nanotech center. The students should apply a project there. Or training also they do it. So it's a center, I'm not getting it immediately. It's comes under EC department.
Or when I'm coming, if you open your lab, then I can give
Sure, sir. That we can definitely, sir. You can do that, sir. Next time when you coming, last time it was very sharp.
We can arrange a session for all the students.
Yes, sir. We can do it, sir. We can do it. Sir. Thank you, sir. Even in YouTube, like if you want, we can do some interaction and we can post it also.
Yeah.
We can do it. Thank you.
Thank you, sir, for your wonderful responses.
Thank you, doctor. Thank you for your presence in BioYojana 2021, organized by TANBI R&D Solution. Your speech was really very, very knowledgeable, and it's a knowledge sharing talk, sir. And a lot of content is there. The slide has the most valuable things. Thank you, sir. Thank you very much, sir. Thank you for your presence.
Thank you very much.
Now, Soundarya, you can give the vote of thanks to Dr. Vijay Shankar Asokan, sir.
Thank you very much, sir, for sharing your profound knowledge with us. Your session was full of valuable information on microscopy and imaging. Thank you, sir.
Thank you, everyone.